Hello, everyone, and welcome to October's Ask the Expert session. I am Mark Chatterley. I work at PSC Support. Usually it would be Paula sat here, our Chief Executive. Unfortunately, she can't make it tonight, so I've stepped in. Today, we have got the wonderful Professor Iqbal from the Queen Elizabeth Hospital in Birmingham. I hope I say this right, a professor of clinical microbiome. See, I did stumble over that. He is a wonderful clinician. He doesn't do clinics anymore, which is a shame for me because he was my gastro doctor. Very useful. So I'm a little bit biased in this, I have to say, but I think you're wonderful. You're going to you're going
That's
to talk
always
to us.
nice. Sorry.
Well, yeah, you got to have a little bit of praise to start these things off. And he's going to talk to us today about PSC-IBD, PSC-IBD, which is, I believe, the best way to explain it is it's kind of the new name for the type of inflammatory bowel disease that PSC people tend to have. I'm guessing that's not always the case, though. But I will pass over to you. I believe we've got a bit of a presentation, and then we can have some questions at the end if you want to fill in the Q&A little taskbar.
Yeah, thank you very much, Mark, and thanks very much for asking me to do this. I have to say that I've been doing the PSC-IBD clinic at the Queen Elizabeth Hospital for the best part of a decade, really.
Yeah.
And I've now retired into research, but I've got only because a very good fellow has now finished his PhD, and he did quite a bit of work on that, so he started. But I have to say that it's the most stressful clinic I've ever done, really.
You have to deal with people like me. That's why.
No, no, not at all. The people and the patients are wonderful, and I really enjoyed that aspect. But it's just because it's so important managing the
Yeah.
IBD as PSC, because I don't really think of PSC and IBD being different. I believe they're intimately linked, and the IBD actually, to a large extent, in a lot of this condition is driving the liver problem. So, and you mentioned, microbiome is my main thing, and so I'm going to not talk too much about the immunology of PSC, but I'm going to focus on the microbiome aspects really. Um, so yeah, what is PSC-IBD? I kind of, can you see these slides? Yeah.
We can, yeah, they're on my screen now, so which means everyone should see them.
Yeah.
Yeah.
Okay. Okay. So yeah. Well, let's start with the gut microbiome. And this is obviously become something of great interest in the last 20 years since the advent of genetic ways of investigating this. Because intrinsically, if you take a stool sample, pretty much all of it, all the bacteria in there will die as soon as you produce it, because they don't live in oxygen. And in fact, this is a this is a may as well go large here. This is the beginning of the Earth 4 billion years ago, and this is where we are now. And until 2.5 billion years ago, the oceans were full of single-celled organisms, bacteria, fungi, the microbiota, and then 2.5 billion years ago, the vast majority of these early life forms died off as a result of the production of oxygen by early plants and also oxygen producing microbiota in the oceans. And so the first multicellular life forms which have been discovered from the fossil record from about 600 million years ago. And you can see this Girona piana is actually one of the earliest in the fossil record of a multicellular organism, and it's pretty much a gut. And there are two reasons really for the development of multicellular organisms, which then led to animals and the rest of the animal kingdom. One was, if you had a load of cells put together, you're less likely to be eaten. And the second is to provide a home for the microbiota, the bacteria, fungi, and other components of the microbiota, which don't survive in oxygen. So this gut, this anaerobic environment for as a home for bugs, is a really ancient, really very ancient thing. And so as a consequence, the relationship between those I'm going to call them bugs because it makes it just easy. It's not
I,
scientifically correct, but the bugs which live in our bowel and our bodies as we've evolved and our immune systems is a really ancient and complex relationship which we're only just beginning to understand. And so just some terms that the microbiota are the microbial organisms which make up the microbiome. So they're not just bacteria, but also archaea, viruses, and fungi. And we know pretty much nothing about most of that apart from the bacteria, really. The microbiome is an encompassing term for the whole community of microorganisms, including their genetic material. So that implies a functionality. It allows you to understand, because we were looking at their genes now, the microbiome is the whole community and what the community can do. And this is a horrible word, which microbiologists and ecologists really hate, but it's become part of the vocabulary. So you'll hear it a lot more. The dysbiosis is a state where the normal microbiome is, first of all, reduced in terms of diversity, and secondly, imbalanced in terms of makeup. So why have a microbiota in your gut and a microbiome? Well, it's very much a symbiotic relationship. So in the context of the colon in particular, nutrient metabolism is really important. So there's a whole bunch of microorganisms which synthesize secondary bile acids from primary bile acids. And this is likely to be very important in primary sclerosing cholangitis and the liver side of this. Although we don't understand at all how it works, this bile acid synthesis and the action of bile acids is going to be very important. And then the anaerobic or oxygen-hating bacteria in the colon are very important for the digestion of the kind of fibers which we can't digest with our human enzymes. And then the digestion of these fibers in the diet provide short-chain fatty acids, which are really important for the health of intestinal cells and have anti-inflammatory properties as well. So that's really important. And one of the problems with the Western diet is we don't have enough of these fibers. As a consequence, our microbiome, anaerobic microbiome, is degraded. And then if you don't have provide the food, the microbiota start eating the mucus layer in the gut. So that's one of the issues really in IBD. Then obviously the relationship between the microbiota, the bacteria, and the host immune system, or the immune system of our gut, and by extension of the rest of our body, is really important. And there's a careful balance which is out of kilter in chronic or long-term inflammatory conditions. And the microbiota are also involved in metabolizing chemicals and drugs as well. And certainly one of the drugs which has been used for inflammatory bowel disease for many years, 5-aminosalicylic acid, nazalazine, does depend on in certain formulations on the action of microbiota to digest the drug and make it available. And if you have animals which don't have a microbiome, so germ-free mice, they have huge problems with the development of their immune system. But also the other side of it is they don't actually develop inflammatory bowel disease unless you put bacteria into their guts. So diversity is really important. And the alpha diversity is a measure of the diversity between two populations. If you look at the microbiome, I'm not going to labor this, but essentially diversity in anything, government, politics, football, is a healthy thing. And once you lose diversity, then things start to go wrong. This is just a sort of simply, this is just to explain how we look at bacteria in the gut. So this is a human taxonomy. Every organism is classified according to which kingdom they belong to, which phylum they're in, what class, what order, what family, what genus, and what species. So we belong to the family of animals. We are the phylum Chordata. We have a backbone. We're mammals and do all the things mammals do, like give birth to children and breastfeed and so forth. We're primates, hominids, Homo, And the only species remaining on the Earth is sapiens, although I'm sure we've all seen a few Neanderthals occasionally, but then they're not really Neanderthals. Homo sapiens is the only species. But importantly, most of the literature in this space, when you look at the papers around it, use a genetic technique which is cheaper looking at this form of, which allows you to separate humans down to family level pretty much. So you can differentiate between humans and gorillas, but it's not until you get down to metagenomic sequencing, which is becoming more common now, where you can look at the whole genetic structure in a system where you can actually start differentiating down to species level. In the gut, we have 65% or a majority of firmicutes, which are they're called gram negative because that's the way they stain when you stain them under a microscope. But these are anaerobic organisms which don't survive in oxygen. And they're important for digesting dietary fiber to make short-chain fatty acids. Bacteroidetes are the other main, again, anaerobic. They don't like oxygen, and they produce, they help in nutrient digestion generally. And then it's only 5% of the microbiota are made up by other phyla, such as the aerobic or microaerophilic. In other words, bacteria which can survive in small amounts of oxygen, and they're generally the bad guys. The salmonellas, the E. coli, the Shigella, the pathogenic, the stuff that gives you diarrhea, which stimulates your immune system.
So
Again,
just to clarify, just so I might have lost myself a little bit there. So if you take a sample from any human's gut at the phylum level, they differentiate into those three groups.
Those three groups largely, yeah. And the majority of them are anaerobic or oxygen, bacteria which can't survive in oxygen. But the ones that like oxygen are the ones that damage the gut. And those are the ones which get overexpressed or they're overpresent in inflammatory bowel disease. And just as a sort of point of reference here, just to show you how early we are in our understanding of all this, if you're looking at a phylum level at a human being, then you can just about distinguish us from sea squirts, reptiles, but no more than that. So again, it's quite a high level data, this. Diversity really matters. So the Baka pygmies are the original, the only sort of rainforest hunter-gatherers now, and they have a very nice, diverse, rich microbiome. If you go to a local agricultural community like the Bantu, immediately the diversity of the gut and the richness is reduced. And if you look at Americans, in particular Western Americans on a high-fat, low-fiber diet, the diversity is greatly reduced and much more prone to chronic inflammatory diseases. And so most chronic diseases, including gastrointestinal disease, are associated with loss of alpha diversity. And if you look across species, so within the primates now, humans have the worst diversity in the gut, and of the humans, then the Western gut, the American gut, is particularly poor in terms of diversity. And this
would that be, sorry, I'm jumping in again. Would that be because we're not exposed to as much of those germs and therefore they don't take hold, or is it a factor of our diet not requiring them to be there?
Yeah, there's a factor of our diet not requiring them to be there.
Okay.
And a lot of studies of people who migrate from one area to another, it's an environmental thing really. So there are many environment factors which affect the microbiome, and diet is by far the most important because that's pretty much maintained through life. So yeah, it's a dietary thing. Thank you for asking. But this reduction in diversity and this dysbiosis, therefore, is a feature now which has been recognized in many chronic inflammatory conditions. You can see from IBD to chronic liver disease, chronic kidney disease, certain brain disorders. You can read them all there. This is an important pathogenic factor. And there's this American gastroenterologist, Dr. Blazer, has wrote this very nice book called Missing Microbes, which explains how these, certainly in mice model way, how important a diverse microbiome is in maintaining health. And there's this clear association in the 20th century between a reduction in infectious diseases mirrored by an increase in chronic inflammatory conditions with the advent of penicillin in 1920, in the 1920s. So in inflammatory bowel disease, which is the paradigm for this really, here on the left here you see a healthy gut with tightly bound cells, a nice thick mucus layer, a diverse, although it's not really diverse here in this picture, but a diverse microbiota living within the gut, and an immune system which is basically tolerant. All the immune cells here are mainly T regulatory cells, which are there they're anti-inflammatory, and they kind of signal that everything's happy. Once you get inflammatory bowel disease, then the mucus layer gets degraded, the cells become leaky, there is a translocation of these bacteria across the cell wall. The bacterial population is marked by a reduction in diversity, a reduction in short-chain fatty acid-producing bugs, an increase in these proteobacteria, these oxygen-liking bugs like Shigella, E. coli, Salmonellas, and instability. And as a consequence of that, the immune system gets triggered off. You lose your T regulatory cells. You get these they're just, I'm sorry about the labels, but these cells actually are inflammatory cells. They cause inflammation in the gut, and they produce all these chemicals against which we now have therapeutic targets. So TNF alpha, IL-20, 12 and 22, which are targeted by drugs, TNF, IL-23, and these inflammatory cells can also target other organs as well. And the paradigm is now that off-target effects, such as joint problems, liver problems, are probably actually programmed from the gut in terms of these T cells, and hence the intimate relationship between IBD and PSC. And now this isn't gospel, but there's a hell of a lot of evidence to just suggest this, and this is what I believe. So the treatment for IBD in PSC is really no different from the treatment of IBD in IBD without a liver manifestation. So basically, we tend to start with 5-aminosalicylic then steroids as a short treatment to kind of bring the condition under control. Immunomodulators such as azathioprine, 6-mercaptopurine, sometimes methotrexate, which generally reduce the effectiveness of these T cells, and then biological agents, which are targeted now as monoclonal antibodies against TNF alpha. So we have infliximab and adalimumab, ustekinumab, which targets IL-23 and IL-12 as well, and then the son of ustekinumab, risankizumab, other monoclonal antibodies, which are antibodies directed against these chemicals, are the mainstay of the treatment of inflammatory bowel disease. And they're very powerful, they work very well, but not for everybody, and the effect does wear off. And then there are some small molecule tablets which have come out now, which have a very powerful effect on all of this by acting on the cell. Things like tofacitinib, and you will have heard of these drugs. But essentially, the treatment algorithm is this. It's basically a top down. But my message really for PSC-IBD is maybe top down it would be better than bottom up. So maybe start earlier, because it's really important the IBD is kept under control. So inflammatory bowel disease is driven then by whether it's PSC-IBD or IBD, it's the same thing. You have a genetic predisposition, which is there have been more than 200 genes associated with. There isn't one gene in most people. This is just the kind of general makeup. There is a disturbance in the microbiome driven by environmental factors, which are poorly defined, but clearly diet is very important. Smoking is another. And this leads to a degradation of the intestinal barrier and an immune response. And that immune response can either be just directed to the gut or the joints or the liver, and PSC is part of that liver paradigm. So ulcerative colitis is without liver involvement, generally affects the left side of the bowel. We all know it causes bloody diarrhea and is a hell of a problem. But it is rising in an epidemic way in the world. So if there are three things which are important, the immune system, our genes, and the environment, the immune system and our genes haven't changed in the last 40 years, but the environment has. So, and this is where change in the microbiome, I think, becomes really quite important. In chronic liver disease, PBC, PSC, autoimmune hepatitis, there is a similar reduction in diversity in the microbiome, an increase in those E. coli, enterococci, which are talking about, which are the those oxygen-liking damaging bacteria, and a reduction in immune tolerance as a consequence. The difference in PSC-associated IBD, and I'll show you some evidence for that in a minute, the bile acid synthetic pathways, the way you metabolize bile does seem to be more important in people who have the liver side of the problem compared to just IBD without liver manifestation. So, at least 80% of people with primary sclerosing cholangitis will have inflammatory bowel disease. Sometimes it's there from the beginning. Sometimes it comes after the liver transplant, and which we don't really understand why that is. And you can see here that it can either be right at the beginning of the disease, or really even up to 5-10 years. Sorry, 5 to 15, 20 years after the disease. The other side of it is that if you look at a cohort of people with inflammatory bowel disease and actually have a careful look at the liver by imaging, you will find 1 in 5 of those patients, people without any enzyme problems in their liver, will have some changes suggesting primary sclerosing cholangitis. And this is it's kind of I don't want to put scary stuff up, but I think I just want to use this slide to just illustrate the point. If you have a liver transplant and you have inactive IBD, you are much more likely to hang on to the liver than if your IBD is active. So it's a really important point in terms of maintaining your IBD under control. It's very important, this. And you can see here from this Australian study that if you look at people who've had liver transplants, then if your IBD is mild compared to if it's severe, again, there's a big difference in liver health here. So, when I first started doing this clinic, my predecessor really didn't take the IBD very seriously, but for me, it's been really important, the IBD side of it, and Palak is always knocking on my door and saying, fix the IBD. So there isn't actually an active liver treatment yet, right, which is recognized for the things that are in development.
Do you know how they are measuring the mild, moderate, and severe? I don't have a kind of a scope for how my IBD would be categorized, if that makes sense. Because to me, it's just horrible, or I can cope with it.
Yeah. So the I think the important thing, Mark, is that you need to look with an endoscope every year.
Okay.
Honestly, and it's not just for a cancer surveillance. It's there to make sure that it's okay and looks okay, because we don't have very many non-invasive markers of IBD. We have fecal calprotectin, and we have with CRP and stuff like that. But the thing is that most people with the form of PSC IBD, the disease tends to affect this side of the bowel, the right side, as opposed to the left side, and so the rectal bleeding and the sort of tenesmus, which is the sensation that you really need to keep going to the toilet, is not so much of an issue. It could just be a general feeling of not quite right, So I think if we have fecal calprotectin, we have to clinical scoring scales, such as Harvey-Bradshaw index for the more sort of Crohn's type of phenotype, where small bowel is also involved. Mayo scoring for ulcerative colitis. But I think that the key thing is keep an active eye on it, like do an endoscope every year or two, you know? I think it's
So
really,
that sort of definition of mild, moderate, severe isn't necessarily something for us to worry about as long as we let you know what the symptoms we're experiencing
that's
and
it. Yeah.
let the colonoscopy and all of that inform your decision making.
Yeah, and we use non-invasive markers such as fecal calprotectin and CRP and so forth. But I think it's really important to keep an eye on it, really,
Yeah.
so much. Yeah. So Nabil, who did a PhD with me, and as I run off to Dubai because of family issues, I mean, it's a real terrible thing, was able to show that in PSC, patients with PSC and IBD compared to IBD alone, compared to healthy individuals without IBD, there's a big difference in the genes which are expressed, and the microbiota actually in the gut as well. And people have shown, certainly in both in IBD with or without liver disease, there's this general picture of a reduction in diversity of bugs and a change towards the more nasty bugs. But there are differences between PSC-IBD and people who have ulcerative colitis without IBD in the expression of the actual balance of bugs. And this chicken and egg question always happens. Is this a causation or an association? Is this something that causes a problem, or is it just a consequence of something else which is happening in the liver in relation to bile acid, which is causing an issue? But yeah, apart from Nabil, other people have found the same thing, that there are differences in the microbiome in PSC compared to healthy individuals, both in the reduction in the good guys here. I'm not going to, which is the Bacteroides and Firmicutes I was talking about earlier, and an increase in the Proteobacteria, Streptococcus haemophilus, the Fusobacterium, the pathogenic inflammatory, and also a difference in the bile acid conjugating bacteria as well. And Nabil was able to show this quite clearly. There's a difference between patients with IBD and PSC versus healthy controls as well. So how do you modulate the microbiome? So there are three ways. You can either bung in broad spectrum antibiotics, so you can subtract bacteria, for example, vancomycin, which I'm going to talk about in a minute. Quite often antibiotics make things worse, particularly for example, C. diff, which I think people will know about, Clostridium oides diffuse infection. The more antibiotics you put in, the worse it can be. Bacteriophages, of which there are more viruses in the gut than there are bacteria, would be a very neat way to manipulate the bacteria in the gut. But our science is not up to it. We still don't know the first thing about this, really. And then diet, postbiotics, so putting extra bacteria, all these stuff you can buy from Holland and Barrett's. There's nothing really effective there. FMT, fecal microbiota transplantation, which is the kind of crazy thing that I do, is the most effective way of changing a gut microbiome, because it takes a whole microbiome and replaces it with another microbiome. And eventually we will get through FMT, through our science, towards rational live consortia of bacteria, which will be helpful, but we're not there yet.